Hello everybody and welcome to the Replace Educational Webinars. My name is Mieke van Mulders and together with my colleague Maud Everaert, we are organizing a series of educational webinars for the Replace project. So we are still waiting for some people to enter the webinars, so that's why we also want to give you a bit more general background information on Replace. So Replace is a scientific project which is funded by the Flemish and the Brussels regions, so the governments who are responsible for the implementation of the animal welfare directives, and it is coordinated by Cienzano and the Vrije Universiteit Brussels. In this project, we aim to collect all available expertise that we have in our country in one central database in order to further stimulate the use, development, and implementation of alternatives to animal testing, also known as new approach methodologies. So for this, we are organizing many different events. So we actively promote and share knowledge on NAMs via our newsletter, our website, our database, our social media channels, but also via the organization of events like this one. So this is already our 5th or 6th series of educational webinars, and we have a second one also planned in April, where we will zoom in on NAMs for DART and neuroscience. So today, again, we will have 2 speakers who will present their work. So before we start, first some housekeeping rules. So every speaker will have about 20 minutes to present his or her work. There will be, after each presentation, 5 to 10 minutes foreseen to ask questions. So don't be shy and put your questions in the chat. We would love to see your questions. If you have need a certificate of attendance, because the webinars are also accredited by the Flemish and Brussels region, don't hesitate to put your name and affiliation in the chat. So my colleague Maud will then provide you the certificate within 1 or 2 weeks after this webinar. So it will take maximum 2 weeks before you receive it. All participants are automatically muted when you enter the webinar, so just so you know, but obviously you can pose your questions in the chat. Also, all webinars are recorded and they are then placed on our Replace YouTube channel, which is freely available and accessible. So if you miss a webinar, you can also rewatch it via this way. And after the webinar, you can also complete a short survey of about 3 minutes if you would like to see a particular topic of interest presented in one of our future webinars, or also if you would like to present your own work, you can also let us know. So also don't be shy to leave your comments. We always try to implement your feedback as much as we can. So that's why also as a second speaker, we will have a very practical essay that is already well known. But first, we will have the presentation of Dr. Anaïs Schaschkow, who is a pharmacologist and a researcher specialized in the life sciences and the health sciences. So she is currently working at Antleron, where she works at the intersection of therapeutic and technological innovations. And she works mainly on developing new innovative in vitro models, such as customizable bioreactors that mimic human bone environments for bone implant evaluation. Which also helps them to reduce animal testing and which can also accelerate the use of NAMs in medical device development. And Anaïs has a lot of experience in cell therapy and biomaterials, where she significantly contributes then to regenerative medicine and therapeutic applications. So Anaïs, the floor is yours.
Thank you. Well, thanks for the nice introduction. And I then don't have to do it myself. This is perfect. So I will immediately share my screen. And then if you can just confirm at Replace that everything is okay.
It's okay.
Perfect. Thanks. So yeah, I'm Anaïs Schaschkow. I did my thesis in France and then I moved here to Belgium, working right now at Antleron as the Replace team nicely introduced. And I will talk to you today about an in vitro model, which is basically the living chamber. So before to dig in a bit more in the subject, I need to put a little bit of context around it and basically explain what are the relevant bone models that we have so far, which are relevant towards clinical application. This is the most important things. Here you will have as a gold standard, I would say, really the large animal models. And the reason why these are the gold standard is because if you imagine the size of human implants, basically they can be transferred directly in the animal without really modifications. So the constraints, the mechanical strengths and everything would be quite similar to what you will have. But major drawbacks, of course, is the cost, the main topic of the webinar today as well, the ethics, right, for animal model usage, as well as the low throughput. So it's not imaginable that you can do millions of tests on large animal models in a lab. So this is for sure not possible. So for that, you could have the use of smaller animal models, such as, for example, mice or rats or even rabbits over there are used. Either with the same drawbacks, which are cost and ethics, you will have on top of that the fact that it's impossible to put an implant that is made for human in such a tiny animal. So usually you will have what we call ectopic implantation and basically put it on a place that it's not at all where it should be. So to completely deviate from animal model, researcher already developed some in vitro models, and we already have quite good ones available. The one and the simplest one would be, for example, all 2D-based model composed of different cells, such as MSC, which presented the advantages of also being from human, so more close to the final application, and can be really mixed with other cells and makes it quite a complex mixes of them. However, the environment relevance is quite limited, as well as the testing possibilities. So researcher also developed more evolved, let's say, models, such as, for example, the spheroids, the hydrogel use, or even the use of microfluidics. This is really cool. However, if you want to really test an implant, as you would do it more in the large animal models, it's quite difficult to do so. So we try to really, with our model, which we call the living chamber, which is the one that you see here on the slides, connected to a medium reservoir and a peristaltic pump to really fill the gap between the research and the clinical needs. And before to explain to you a bit more what is really the living chamber, I would like to come back to why did we develop this living chamber, basically. And this was using our smart bioprocess platform that we developed at Antleron. And the first step that we have over there, usually it's, okay, connect to either researcher or ourselves, because we are also a researcher actually, and say, what are the pain points we will need to solve, or what do you really need? In this case, so the problem statement and process analysis, we know and we discuss with people saying that the final stage of differentiation, mean if you differentiate your cells towards bone, the bone is hard to obtain in vitro. As well as to really test, as I explained, an implant, it's quite hard to have an efficient method for testing it. So based on that, we went to our first step, let's say, or first engineering package, let's say, which is composed of simulation, design, as well as 3D printing. For that, we have simulation tools, CAD design tools that allow us to really, let's say, put our ID into shape and to do some first testing to see which one could be the best. After that, we would print physically the device or the bioreactor in this case, and then, of course, experimentally testing it. Once we have that, we will do iteration to correct some technical details most of the time and have something that is as user-friendly as possible. Once we have that, and when doing the experimental testing, we will also try to put next to the process what we call a digital twin. A digital twin is really a twin of your process that can be run in silico, which will then basically help to reduce the number of experiments you will need to do. So on top of reducing animal usage, it also helps you reduce the experimental load that you need to do. And next to that, we also are on the way to develop soft sensing technologies. So basically, what is that? It's kind of a proxy that can see or reflect the wanted readout. So for example, here, if differentiation is checked, you will have to measure something without destroying your sample that will tell you yes or no, your cells will be differentiated. And then we close the loop, and either we go through a new iteration if the result is not satisfying enough, or we will end up with a relevant and satisfying model, which I hope you will convince of what I will present to you today. So to come back a little bit to the living chamber itself, this is the design of it. So you will have a chamber over there, a lid and a fixation ring, which are the technical part to contains everything. And the smart part, as we like to call it, is this two-part scaffold. Why? Because imagine, so we are in this bone context, right? And we have in our outer scaffold cells seated on it. In the middle, we will put like a full plug, so something that will really clog the middle of the scaffold. After that, we will run cell differentiation and obtain, hopefully, a bone-like environment. Once we have that, we can then remove this mock-up plug and replace it with the test implant, and later on run biological evaluation. And this biological evaluation is very similar to any other readouts that you could have in the lab, because I tested a lot of them and most of them are basically compatible with the system. And it's also nice because it's what we call a high lab scale throughput. We can put up to 24 replicates on a perfusion pump at once, and you can multiply it by number of pumps if needed. So beside the full principle or the idea, we also needed to basically validate that what we expect to work will really work. For that, I will present to you now the first experimental study plan we did. And this was to show the osteodifferentiation in static conditions. So these are the conditions that are mainly used right now in research for in vitro models, versus our living chamber condition, which is done in perfusion. First, we used bone marrow derived TERT MSC, so immortalized mesenchymal stem cell that we immortalize ourselves to have our own model. And we put them on our scaffold, so same scaffold that is in the chamber, but basically outside culture statically in a well plate. We culture them for 21 days in an osteo medium, which is a medium that basically contains all the cues normally that cells would need to go towards the differentiation. And then evaluate what do we have at the end. Then we also use, of course, the same cells on the scaffold in the perfusion device, but here we use two different media, either maintenance one, so very regular one that you will use to just amplify your cells, or also the osteo medium. And again, check what do we have at the end. The different readouts we checked there are either metabolic activity, gene expression, mineralization, ALP activity, and we also did some CT scans. And the full goal of that was to be able to validate that the first assumption was correct. Basically, we can achieve bone-like environments. And on top of that, of course, we included some 2D controls, so for TCP, tissue culture plates, to serve as a reference. So the first one I would like to present to you today are the metabolic activity during the osteodifferentiation. The one metabolite that is presented here is lactate. And we can say that basically, in a very simple words as well, the more cells you will have, the more lactate production you will have. And this is the case when cells are like 3D proliferating or just dividing normally. So if you check on the graph on the left, here is really a TCP case, and it's interesting to see when you use osteo medium, so the one that contains the cues to differentiate the cells, the metabolic activity of the cells is changed. So it's really lowered in osteo condition. It's the metabolism is then affected. Then if we checked on the graph on the right, here you have the dynamic osteo condition and both the static osteo condition here and the two dynamic one in perfusion. What is interesting to see is like compared to the static one, in which the cells still produce a lot of lactate, having the perfusion set up, whatever the medium, reduce this amount of lactate produced. So it means that apparently just the perfusion could already trigger the cells towards the differentiation pathway because they are not really prone to proliferate that much anymore. In order to confirm that, we checked another metabolite, which is not one that you will have, let's say, the use to see, or at least I don't think so, which is glutamate. And it came from, of course, literature study, but also some internal observations. And we've seen that in TCP, for example, when we will culture the cells within the osteomedium, we will have an increase of the glutamate production. And we were like, okay, maybe that's a good thing to follow up. And then we did the same measurement on the static versus dynamic in the living chamber. Here are just both with the osteomedium. And you see a very drastic difference in the production of glutamate, which is really, really increased. So here it seems that the increase of glutamate is also showing a better differentiation over there. And if you remember what I said in the first loop slide, the soft sensing development, this is also something we are doing over there. And the use of glutamate to basically follow the course of your differentiation, this is a very good example of a soft sensor. And then you can use it without sacrificing samples. So this is also reducing the number of samples you need to run your condition and reduce your experimental costs. Then if we go back to something a bit more classical, if I could say, here are the gene expression that is in control on osteomedium. And on the top of the slide here, you have a little scheme presenting the course of osteodifferentiation and a bunch of different genes that are expressed all over. And here the final stage, which is osteoblast and osteocytes. Interestingly, if we check most of these genes over there, so here you have the comparison between the perfusion in the living chamber in the regular medium, so without any osteocues, compared to TCP. And here we can already see that most of the genes are the tendency to increase without really being significant, but it seems that there is something like kind of a trigger over there. Then we compare it to really the osteo conditions. And here you see that here, yes, almost all of the genes became significantly higher. So mean that adding the osteodifferentiation cues on top of the perfusion effect really increased the differentiation potential of the cells. Then I would say the most important part over there, or the one we really want to tackle as a pain point, is this final stage of differentiation, this mineralization, really the bone formation achievable in the model, yes or no? So this is this final that is depicted here. And for that, we use alizarin red staining, which stains for calcium, and as well as ALP, so it's alkaline phosphatase, which is really a bone-specific enzyme. And you have some little pictures over there in the middle as well of the different scaffolds and cells on top of it. And what is nice to see, it's as just shown before, right? The control condition, even in the maintenance medium, they already kind of trigger the cells towards differentiation. We already have some mineralization over there and some basic ALP activity. However, if you really add the osteocues on top of it, then you really maximize the outcome of the model over there, and it's really needed to achieve a better differentiation, as nicely shown as well here in the picture. On top of that, we did a very cool thing, I would say, which is CT scans. So this light is quite heavy, so it might take a little bit to start playing, so I hope you can see it nicely. The CT scans are basically X-ray and will only reflect a very dense tissue. So same like X-ray will show your bones. Here, it's exactly the same principle. Here on the left, you will have the scaffold support. So this is the two-part scaffold normally, which is not visible in the scan because it's made of polymer. So it's fully transparent to the X-ray. Then you will have the maintenance medium over there. So it's really without the osteocues, and you see some little spots here and there, also proving that we have the start of a differentiation, but not really. But then if you look at the one with the osteomedium, it's really clear that we have a nice mineralization over there and the formation of bones all along the scaffolds. So I would say here really put the stamp on, yes, we can create this bone-like environment in the living chamber using the perfusion setup as well as the osteomedium. Then to prove that the idea of having this implant testing is also working, we wanted to see if putting the implants inside is working as we would like to. For that, very simple experiment was done over there. We put cells in the outer scaffold with the full plug in the middle and did some culture. Then after some days, we replace it with different kind of implants, which could be either titanium implant or polymer in this case, with different porosity, and then we culture them further. On that, we checked metabolic activity, viability, as well as cell ingrowth. And this was to validate this two-part scaffold principle. Before to really be able to test it or to run the biological validation, what we do first is to check in silico, let's say, what the porosity of the implant could influence on. Because here, if I say porosity is 50% versus porosity of titanium implants is, for example, 90%, it means that in the titanium case over here, only 10% of the full volume is material. The rest is air. Whereas in the other one, it's 50-50. So to see what this influence can be on our or in our bioreactor living chamber model, we did some CFD, so computational fluid dynamics over there for checking fluid flow, oxygen distribution, as well as nutrient distribution. And I would say not surprisingly, we observed that with the high porosity, where a lot of empty volume is present in the middle, you have more fluid flow going through here than in the other one. But the most important thing is like the oxygen repartition as well as the nutrient distribution is not affected by that. And so we don't expect any trouble changing porosity in this range that are depicted there. So this is a little picture to show you. So after a couple of days, we reopen our chambers and the cells were already cultured here around. We remove the full plug and replace it with an empty test plug, which here, the one on the picture is titanium, and then we culture it further. Here we see the cumulative lactate again, so the first metabolite I presented to you before, and so the more lactate means more cells, as I said as well. And you can nicely see that the lactate production is increasing over time, even after the implant insertion, so that's really cool to see that the living chamber can successfully support the use of this implant, whatever the material or porosity. You will see here that the titanium is a bit lower than the other one, but this is quite easily explainable, and I will also show you in a more visual stage how it looks like. Here we did calcein staining. So calcein is staining basically in green live cells. And here you have different titanium implants, which are different print parameters, but basically both of them are titanium. And in the middle, you have the polymer one. What is nice to see is here is the limit between the two, and you nicely see an interaction with the cells that were here first and completely colonized or ingrowth into the empty implants. And remember, the lactate was a bit lower for the titanium. This is purely due to the available surface, which is only basically 10% of the full volume, which is also the reason why here cells needs more time to fully colonize the scaffold over there. So with that, we can put the second stamp and saying that this two-part scaffold principle is validated. After that, I also wanted to show you a little bit more how it looks like in our lab. So you have like a zoomed in version over here, and that's a picture of a full pump. So it's 24 samples run at a time, and their medium reservoir connected to it. Which is also nice with this model, and because we have the full, let's say, freedom on the scaffold, the bioreactor, as well as even the holders, because we are doing them ourselves also. It's like you can adapt this to the needs. So these are, for example, the standard setup, the two-part scaffold. You can have only one big scaffold inside. You can also test ceramics, for example, or as shown before, titanium implants. And as part of the consortium that we are also working with, they asked us before to test a big chunk here of titanium first, which then we had to adapt the scaffold for, as well as the chamber, and basically everything is possible to suit the needs. As a conclusion then, I would say that the living chamber is an efficient and robust lab test platform for implants. It's suitable to test different parameters. timing and also type of readouts, quite similar to what you can do in daily lab life, I would say. It's also fully customizable, as just referring to the previous slide, and can accommodate different implant types. But you can also imagine running a completely different experiment with like different timings or implant insertion, different cell type in the middle and in the outside. So it's clearly modular, let's say. Of course, the best thing is I hope it will help to reduce, even if we dream about it, replace, but it's going to be complicated, but let's already aim for reducing the need of in vivo testing, and how it's basically by pre-screening in a better and proper environment. So then the need for the final animal testing will be already reduced. And I hope that I succeed to convince you that could also be used to evaluate other things, such as, for example, drug or therapies, or also adapted to other medical innovation and develop, as shown with the glutamate over here, soft sensing technologies. With that, I would like to thank the Prosperos Consortium, which is basically the one for which we start to develop this model, and we're still continuing and improving it at the moment. And also you for your attention, and I'm really happy to take any questions right now. And if you want to contact me, here are my details and also a QR code to our website if you're interested to see what we can do as well. So thanks a lot, and happy to take any question.
Thank you, Anaïs, for this nice and interesting presentation. While we are waiting for some questions, I actually already had two small questions. So first, I was wondering for which disease areas did you have the in vitro model developed?
So this was basically for personalized implants, like generation. So in the consortium, what they ask is they want to really, so right now with the 3D printing, right, we can really put the shape according to the patients, and we can also put any treatment or coating on top of it that will then be towards the patient. So this was the goal over there, to be able to test really in advance with patient cells or anything, the use of the material that is intended to be used later.
Oh, yeah. And because you mentioned that the 2D scaffold was already biologically validated, I was wondering what are then the next steps if you want to bring it more closer to clinical trials and patients? What are the next?
This for us, so the in vitro model is basically set as, okay, we succeed to prove that this is working. Right now, we're really waiting for applications as well, and for people showing that this model can replace basically the animal model. That's really one of our next step. It's to compare both and to see if the discrimination power is as good as what you can have in the animal model, for example.
And is this also then incorporated in the Interact project itself, or is that then for a following?
No, it's not exactly for this project. Then this project is more towards the clinical application and not really the model extra validation. But well, it's already the second part of this Interact project, so it might be also a third one, which then could include it maybe.
Okay, and when is the project ending? The Interact?
2026.
Oh, yeah, okay. Okay, thank you. So I don't know if there are other questions in the chat.
There is one question in the chat. Sorry. Have you already applied it to patient stem cells? And can you include screening for possible allergies as well?
So we didn't do it because we don't have access to basically patient materials on a daily basis, which would be very cool to have. So if you have any clues, I mean, please contact me. I'm happy to take that. And I think it can indeed be used for screening anything which is patient related. So if you would like to test any reaction of the patient cells to, as right now, allergic components, so you could probably test it in there. Maybe the model, as I said, should be adapted. You don't need this two scaffold part, or maybe you will need it a bit differently. I don't know, putting the patient cells outside and then the allergen inside that you will put then in contact with. I think we can think about something that can work indeed for that.
Linked to the fact on patient stem cells, I saw that UZ Leuven is a partner. So isn't there a possibility?
I think, well, the partners that we have there are more really towards the, so there is multiple facets, I would say, in the Interact project over there. And there is also one which is more using brace to correct some bone defects over there. And they are more on this part. So it's probably a possibility, but then, yeah, maybe not now, I would say, sadly.
All right. And I'm not sure if I grasped the answer to the second part, if you can include for allergies.
Yes, that's what I said. I think you can include it, but basically it needs to be adapted, and also the protocol needs to be then sent to see if it's feasible to do so.
Okay, thank you very much. I don't see any other. We have one final question from Yuri. Have you thought about using this approach for horses, which are also often patients with bone or cartilage defects?
Yeah, actually, we think about it. We already think about broaden the application, and what we used already horse cells in another project, if I can like said, and we try to expand them to also have horses MSC, and indeed differentiating them or testing different treatment over there would be quite interesting. So I think it's a very good point that you raised here as well, to use it for also the race horses and everything. This might be super interesting indeed. Yeah. I think it was a second part of the question, but I didn't see it, sadly.
We did have a third question. If you use the tests, the interface of material and bone cells.
Yeah, here not, because we are waiting for our partner to really tell us what they want to test, basically. And so what we tested right now is to see if there is any interaction. And what you could do is imagining at the interface to really separate both and do imaging or any kind of stainings over there to see how this communicates or works together.
Yeah, and in particular for the evaluation of osseointegration?
Yeah, this I would say maybe the CT scans are the best over there, because it can really show you the different parts, and if you have fusion or not in basically what you had previously, the bone-like environment that we developed and the one you would like to create. So indeed, next step would also be to use osteoconductive as well as osteoinductive materials to be tested in there, and to be able to evaluate it in a proper way.
Okay, thank you very much for your detailed answers and for your very nice presentation again.
Thank you.
So then we can go to the next speaker.
Yes. So the second speaker of today is Marie Pardon. She is a PhD student at the Katholieke Universiteit Leuven in the Laboratory of Pharmaceutical Analysis and the Laboratory of Molecular Biodiscovery. She graduated with a bachelor in Pharmaceutical Sciences and a master's degree in drug development. During her master thesis, she started working with zebrafish models for neurological drug discovery. But currently she's doing a PhD, and her PhD project is focusing on developing liquid chromatography methods for identification of organic micropollutants in wastewater, in addition to ecotoxicological testing using zebrafish. And recently, Marie also won the Beltox Best Oral Presentation, so we are happy to have her today to present her work.
Hi, thank you for the kind introduction. I hope everybody can hear me well.
Yes.
Okay, perfect. Then I will also start sharing my presentation. Okay. I hope everybody can see the slides.
Yeah. Yes.
Okay. Yes. So indeed, my name is Marie. I'm a PhD student at the KU Leuven, and today I will give like a brief introduction to zebrafish embryo assays for toxicology, ecotoxicology. So a bit short overview presentation. So first, I will start a bit with an introduction on the zebrafish model. So the zebrafish are actually a tropical freshwater fish, and they're characterized by these typical striped patterns. That's why they're also called zebrafish, of course. These fish have a very small size, like up to 4 centimeters, and they're very easy to keep in captivity and very easy to breed. So that's why it's also very useful as a lab animal. Originally, these animals occur mostly in countries like India, Nepal, Bangladesh, and they were also first described during the British colonization area of India by a Scottish physician. And their Latin name, the Danio rerio, actually is derived from the Bengali word Dani, which translates a bit to of the rice fields, and this relates to their natural preference for like shallow pools and shallow rivers. And then a bit more about their history as an animal model. Actually, the zebrafish research already started in the 1960s, but they became famous due to this scientist called George Streisinger, which was a tropical fish enthusiast from the University of Oregon. And back in those days, the main animal models actually used to study development were the fruit flies. But this George Streisinger, he was actually looking for a vertebrate model to study a bit development. And he actually highlighted as one of the first the potential of the zebrafish model. And afterwards, it really attracted the attention from a bigger scientific community. And this is because the zebrafish have a lot of advantages as an animal model, such as their small size, external fertilization, and very fast development. So it's very ideal to study development compared to other typical models, such as, for instance, the mouse model. And then an important other historical step was the Zebrafish Genome Project. This was since the early 1990s. There actually were these genetic screens to search for interesting mutations in the fish. And here they could observe like, there are some interesting effects, but it was very difficult to identify like which gene was now actually causing which effects, because the full genome sequence back then was still unknown. So this started the Zebrafish Genome Project. It's actually performed by scientists from the Wellcome Trust Sanger Institute, and they were able to fully sequence the zebrafish genome. This was a very big project. It took about 10 years, but due to this full sequence, they were able to find that actually about 70% of the human genes have at least one similar gene in zebrafish, and even about 48% of the human genes that are associated with a disease, for example, can also be studied in the zebrafish. So yeah, this project was really important because this gave a very useful reference tool for scientists, as the same with other model organisms. And so in addition to this genetic similarity to humans, the zebrafish also have many other advantages to use as a model. For example, it's very easy to do genetic manipulations, such as, for instance, CRISPR-Cas methods on this fish model. They're also a vertebrate model, and as I said, many of the drug targets in humans also have their counterpart in the zebrafish. Other advantages are that they are permeable to compounds, so it's very easy to study the effects of different compounds, to study toxicity or drug discovery. Their small size and fast development also makes them a very high throughput and cost-effective model. High throughput, you can, for example, use these 69 well plates and then put a fish or larvae in each well, so you can, in a very short amount of time, get a lot of data. And they not only have like a very fast development, but during their early life stages, they're also transparent. This again makes it easier to study the influence on development, for example. And so all of this combines really shows that the zebrafish really is important also for the 3R principle, to replace and refine and reduce animal experimentation models. So actually it's an ideal model for a lot of different applications, not only developmental research, but also in things like drug discovery or ecotoxicological applications. Then a little bit more about this 3R principle, as it is also the topic of the webinar today. Here you can see a short overview of the development of the zebrafish. So in actually a few hours or days, you actually have a development from fertilized egg to already these small zebrafish larvae. And actually during the first 5 days of their life, of their development, they don't require any independent feeding, and they're also not taught to experience any pain or suffering. So up to these 5 days post-fertilization stage, they're actually not considered or regulated as laboratory animals. So this is really in line with this 3R principle, especially the replacement. Of course, there are also a lot of studies using zebrafish also at, like, for example, the adult life stage or juveniles. But yeah, as this is not within the scope of today, I will mainly focus on only the embryos and the larvae up to 5 days, because we also submitted some of these methods into the replace database. So I'll start with the most famous one, the most used one. This is the fish embryo toxicity test. It's actually to determine the acute toxicity of chemicals. This is actually an official OECD test. So you can also find it online, and there's specific guidelines that you have to follow to be in line with this test. So you can expose your newly fertilized zebrafish eggs with your compound of interest for 96 hours. And then every 24 hours, you look at some observations related to the lethality. So for instance, the coagulation of fertilized eggs or the lack of a heartbeat. And then at the end of the test, you have some idea of the acute toxicity of your compounds, and you can calculate LC50 values. But as I said, this is an OECD official test, so the experimental conditions have to be very strictly controlled, such as the temperature, the number of embryos that you test, the different time points. So in practice, this sometimes makes this test a little bit more complicated. There also exist some variations to this test. For instance, you can look at the teratogenic index. So here we not only look at factors like mortality, but you can also look at the malformations during the development of the zebrafish embryo. So you also expose them from 4 to 119 hours post-fertilization. And then instead of only looking at the lethality, you can also look at other factors such as skeletal deformities or motility of the fish. This you can see, for example, in the image here on the right. On the top, you see a normally developed zebrafish larvae, and at the bottom, you see some clear malformed larvae, such as skeletal deformities or even edema. And so after the test, you can actually plot 2 curves. So one showing the mortality and one the malformation amount. And then based on this, you can actually calculate your teratogenic index. And an example of this test was recently published by our research group. So here they focused on a lot of toxic endpoints from metal shredder waste from different sites in Wallonia and Belgium, because this metal waste is, they know there are many different types of toxic compounds present in this waste. So it's interesting to investigate what types of effects could be present. So here they determined the teratogenic index of different types of samples, such as fluff or dust or sludge. And here, for example, they saw that the highest teratogenic index could be found in typical fluff samples. And this could be correlated to the presence of some typical contaminants like PCBs, PPA, or nonylphenols. So this study highlights again that, yeah, it's important that this type of waste is properly managed so that you don't have these types of effects in the environment, for example. Another different important aspect in zebrafish experiments is the route of exposing your embryos. The most popular exposure route is via simple water immersion. So you have just your well plates, for example, a 69 well plate, and then you can put a zebrafish larvae into each well and then just dissolve your compound of interest into the test medium. And then the compounds are taken up by the zebrafish via, for example, the skin or ingestion or via the gills. And so yeah, because this is the most easy to use, it's also the most used. But you also have different exposure routes, such as different injection techniques. You can inject your compounds intraperitoneally or into the yolk or even into the heart. For this, you need a more complicated setup. And of course, yeah, it takes more time, it's more complicated. So that's why it's not used as much, because it's also not that high throughput. But for some compounds, in this way, you can make sure that your compound actually enters the zebrafish and that you don't get any false negative results. Here you see an example. You can also find more information because it's also submitted in the replace database. So here we looked at, we injected actually different fluorescent compounds into the zebrafish, and then we compared a bit the administration via immersion or via different injections. And we looked at both hydrophilic and lipophilic compounds. And here you can see some clear differences. For example, if you use the immersion technique, when you have a lipophilic compound, you can have a nice distribution and uptake of your compound across the entire zebrafish body, as you can see here in the fluorescence. But for some hydrophilic compounds, we actually saw that the uptake via immersion is probably not that high because we don't really see any fluorescence in the zebrafish body. But for the injection methods, we saw that it's already much better. So yeah, it's really the uptake and the distribution of compounds, yeah, really does not only depend on the exposure route, but also of the properties of the compounds. Not only, for instance, the polarity, but also the size can play a role. And so you always have to keep this in mind when you do your toxicological test, that you can have a false negative result because the compound simply may not be taken up into the zebrafish body. Another example, which is also in the database, is also a bit related to this exposure problem. Here we focused more on in vitro metabolism. This is because when you use these zebrafish at a young stage, like the embryos and the larvae, the liver is usually not fully developed. So actually these larvae have a reduced capacity to metabolize compounds. And this again can give the risk that you have a false negative result. So in our group, they developed a protocol to actually metabolize compounds before you expose your fish to your compounds. For this method, they used the microsomes originating from a rat liver. So they do express all of these metabolic enzymes. And here you see an example of some results. So here they focused on parathion. It's an insecticide, and it's known to have like very toxic effects on all types of different mammals and animals. And the main reason for its toxicity is actually caused by its metabolite, paraoxon. But this metabolite is only formed because of the CYP enzymes present in the liver. And so they tested it on the zebrafish embryos. And when they just incubated the embryos with this parathion, but without the exposure to these red liver microsomes, actually only limited adverse effects were found in the embryos, which was, yeah, what you wouldn't expect because it's known to be a toxic compound. But when you look at the microsomally activated parathion, we did see like very pronounced toxic and lethal effects. And this is because it was converted to this toxic metabolite. So here we can conclude that, yeah, if you just expose the embryos to the parent compound, because they don't have the metabolic capacity, you actually have like a false negative outcome. So this is also something that needs to be kept in mind, or that you can try to deal with by using a protocol like this one, for example. And then in addition to these more general toxicity assays, like the embryo toxicity or teratogenic index, you can also have more functional assays for like, if you want to look at more specific toxic effects like neurotoxicity. This is something we have a lot of experience with in our research group. A first approach to do this is to look at the locomotor activity of zebrafish larvae, because after the embryos are hatched, you can have these small larvae, and then they already show these free swimming behaviors. So you can actually expose your zebrafish larvae with your compounds of interest, and then you can use a recording system with like a camera, for example, and then you can measure the behavior of the zebrafish larvae over time. For example, in our lab, we do a lot of research for new epilepsy drug discovery, for example. And here you can see the behavior of a normal zebrafish control larvae. So it moves around a bit, but not very much. And then on the right here, you see a larvae that's been exposed with a epilepsy or seizure-inducing compound. And here you can see that it's really much more active and swimming around a lot because of these seizure-like activities. And in addition to this locomotor activity, you can also look at, for example, the local field potential. This is a method similar to the EEGs in humans. So you actually place a recording electrode on the head of the larvae, and then you can measure the electric signals generated by the brains. And here you can see also some typical epileptiform brain activity when you expose the larvae to epilepsy-inducing compounds, for example. And in addition to, like, for example, neurotoxicity, you can also look at specific organ toxicities, such as the cardiotoxicity. For this, you can really accurately monitor like the heart rate of the embryos, or you can also look at other organs, such as the kidneys. And for this, mostly these fluorescence-based zebrafish models are used. And then finally, I will also give an example of some neurotoxicity assays. So this is what I'm doing right now in my PhD. So we're focusing on these organic micropollutants. Those are various types of compounds, such as pharmaceuticals, personal care products, pesticides, that are all present in our wastewater at relatively high concentrations. And our current wastewater treatment technologies are usually not able to remove actually these compounds from the wastewater. So after the treatment, they most of the time just end up in the environment, where they can have many different effects. And so the goal of our study is actually to look both at the toxicity using, for instance, models like the cyanobacteria, but also the zebrafish larvae, and then combine it with chemical analysis using liquid chromatography coupled to high resolution mass spectrometry. And then we want to see like what is maybe the link between the chemical composition of these samples and also their toxic effects in these different target organisms. So to test this, we actually expose our zebrafish larvae for 24 hours with the extracts of this wastewater treatment plant effluents. And then at 5 dpf, we do a behavioral and visual evaluation. And here for the behavioral evaluation, we expose actually the larvae to these dark light transitions. And this is because when you expose the larvae to a light to dark transition, normally they have like the reflex to increase their total mobility when it goes from light to dark. This is usually some kind of fight or flight response. And we saw that when you expose them to your extracts, usually they don't really show this response anymore or in a reduced manner. So this is clearly indicative that this natural response that they have to the trigger is being reduced. So it's indicative for some neurotoxic effect. And so we exposed them to different effluents from different locations of wastewater treatment plants, and we saw some differences depending on the location. And now the next step will be to actually link it with the chemical composition of these samples, because there are some neuroactive compounds present in there, such as antidepressants or anticonvulsants, which could be responsible for the effects that we observed. And so now finally in conclusion, so a bit an overview. So the zebrafish embryo is really gaining popularity as an alternative model to animal testing because it has so many important advantages and it's useful in such a wide variety of applications. You can also see that the number of publications is also rising every year. So it's really becoming more and more popular. You can look at some general toxicity, such as the fish embryo toxicity or the teratogenic index, but you can also look at more specific functional assays, such as organ-specific toxicity or neurotoxicity. But when you do these zebrafish-based embryos, you have to keep in mind the exposures, such as the immersion or the injection. And then there's also some other factors that can influence the bioavailability of the compounds, such as the metabolic capacity that I mentioned. And finally, maybe also important to keep in mind, so they do have a high genetic similarity with humans, but yeah, of course, their performance is not always equal to, for example, mammalian animal models, which are, of course, still even more similar to human models, but still as a replacement, it can really help in reducing the amount of mammalian animal model testing that has to be done. Okay, thank you for your attention. I hope you enjoyed the presentation and I'm happy to answer any questions that you may still have.
Thank you, Marie, for the nice presentation. It was very clear. I have a quick question before we check the chat. I was wondering why are you focusing? Is there a particular reason why you are focusing on neurotoxicity for the testing of micropollutants?
Yes. So yeah, we don't only look at neurotoxicity, but also some general toxicity. For example, for some wastewater effluents, we also saw some lethality or some other malformation effects. But mostly the samples that I'm working with at more lower or environmentally relevant concentrations, usually you don't really see these lethality or developmental effects. And then it could be interesting to look at these neurotoxicity, because that is what mostly happens or seem to happen at the environmentally relevant concentrations.
Okay, thank you. I was also wondering, because you said that depending on the way the compounds can be absorbed, so it can be via the skin or other ways. So you said that it could induce a bias. So is there a way that you can predict this, that you know in advance that this can be due to the bias of absorption?
Yeah, I think for this, we have to look a bit at the general chemical properties of the compounds, like the polarity, like it has to be polar enough, but not too polar, or it cannot be, the molecules cannot be too big. So it's actually a bit, sometimes the similar rules that apply to like the rule of 5, like what are compounds that are properties to be good drugs, for example. So in this way, you can kind of predict a little bit like, yeah, these compounds will be easier to be taken up than others. But to be absolutely sure, you can always also measure like the actual uptake. For example, using LC-MS methods to see how many of the compound is now actually taken up in the zebrafish. I think we even have one study where we looked specifically at the zebrafish brain and actually measured how much compound was in there. But I think in general, it's sometimes a bit difficult to predict.
So it's basically possible that some compounds, some existing compounds cannot be tested because they don't have the properties to be absorbed.
Yeah, I think maybe they cannot be tested via like the water immersion, for example, but then you can always switch to other techniques such as injection. But yeah, this one is a lot less high throughput. So it all has its advantages and disadvantages.
Okay.
Yeah, I also actually had two small questions. Because in the beginning of your presentation, you mentioned that it's used for acute toxicity testing, and I was wondering if it's also used for chronic toxicity.
Yeah, I think you can use the zebrafish for this as well, but then you have to expose them for a longer time, or and I think then, or even when they're juveniles, for example. But then they're not considered. Yeah, then they're considered as an animal model. So yeah, I think.
And then a last small question, because we actually had a discussion on wastewater during lunch, and we were actually wondering if is your model also used for endocrine disruption?
For this, not really, but I don't know if you ever heard of, for example, the medaka fish.
No.
So you have like the zebrafish, and you also have other fish models, and one of them is called medaka, and those are usually used for things like endocrine disruption because they're very sensitive to endocrine disrupting chemicals and things like that.
Okay, thank you very much for the useful information. I don't see any other questions in the chat.
No.
Yeah.
Okay, so that's the end of this webinar, but we would like to thank the speakers a lot for their presentation. It was very nice. And thank you as well to the attendees for listening to these presentations. If you have any feedback or comments you would like to provide us, don't hesitate to complete the survey, which will be available when you close the webinar. Our next session will be organized on the 24th of April. But if you would like to receive information, don't hesitate to register to our newsletter, so you can do it via our website. If you requested a certificate of attendance, we will provide them in the coming weeks, so don't hesitate to put your name and email address in the chat. And that's it from our side. We hope to see you during the next session. Bye.